This is Algal Sample Processing, lecture for ENR4345, Methods in Aquatic Ecology course at The Ohio State University. In this lecture, you will learn the best practices for holding periphyton and phytoplankton samples in the field until you return to the lab. Finally, I will quickly go over the filtering techniques that we will be using in the next lab period. After periphyton and phytoplankton samples have been collected, it is good practice to keep the samples in the dark and on ice until you return to the lab to process them. This is to slow down any environmental changes that could occur in the sample water between the time when it is collected and when it can be processed in the lab. Filtering of algal samples should occur as soon as possible after collection, and the filters are kept frozen until we are ready for analysis to occur. This is a photo of the vacuum array that we will be using in class to filter our periphyton and phytoplankton samples. We will be using Wattman brand GFF filters, and your instructors have already pre-treated the filters according to the lab protocol. We will be adding one filter to each filter flask on the array, and we will slowly pour the sample into the top of the filter flask to be filtered. The vacuum pump will help pull the water through the array, and anything larger than the pores on the GFF filters will remain on top of the filters. One easy way to think about this is that it's similar to a coffee filter, where the water is able to pass through the filter, but the larger coffee grounds are stuck to the top of the filter. In this lab, we will be analyzing the algae on the top of the GFF filter. However, it is also possible to study the filtrate, or the liquid that passes through the filter, which contains particles small enough to pass through the filter's pores, such as bacteria, as well as chemicals and minerals that are dissolved in the water. After filtering these samples, we will be freezing the chlorophyll A filters and placing the ash-free dry mass filters into the drying oven. During the next lab period, we will analyze the periphyton and phytoplankton samples for both ash-free dry mass and chlorophyll A. Both next week's lectures and the Algal Biomass Laboratory Instructions document in Carmen will have more information about this process.